So today we are here for thesis defense of Mohammad Mazharul Islam. Mohammad hails from Bangladesh, and he went to BUS, or it's called Bangladesh University of Engineering Technology, for his bachelor's. Interestingly, that was also my alma mater. He got his bachelor's in chemical engineering, and then he worked there for a while, and then he moved to Penn State in 2012 to get his master's. And he joined the same group from which I got my PhD. So when I graduated, I think 2 months before I graduated, he got his master's, and then he started working with someone else. Interestingly, that didn't work out, and I got a position here, and then I asked him, why not you come and work with me? And that's how things started. So it has been a journey, I would say long journey for him. But I think he did well, which he will probably show you today. So today what he will do is basically he will give you probably a 45 minutes recap of the work that he did in the last almost 5 years. He worked in different areas. He worked in microbes, microbial communities, diseases, and plants. So yes, it's almost like, it feels surreal that I had a PhD defense last week, and now there's another one. So that means something is going right. All right, so without further ado, Mohammad, take it away.
Thank you for the kind introduction, Professor Saha. Good afternoon, everyone. As Professor Saha has already mentioned, I am Mohammad. I'm a PhD student in his lab, and I joined UNL in 2016. And for the last couple of years, I have been doing my research on metabolic modeling and analysis of living systems. And that includes single organism systems and multi-organism systems. So thank you for being here for my talk. I'll give you a brief overview of my PhD research and show some future directions that are in my head. Now, before we go into the details, let's see. Before I go into the details, these are the publications that have resulted from my PhD research here. In addition to the articles and the book chapter, I also have a couple of manuscripts in preparation. So my talk today will, in essence, give the take-home message from all of these books. I will start with a brief overview of metabolic model development and analysis, and then I'll briefly describe each of my research areas, and I have broadly categorized them in 3 different types. One is single-cellular organism, or microbes, and then eukaryotic organisms, especially plants and human, and then I'll go to microbial ecosystems, which are multi-species systems. So let's start with metabolic model development and analysis. The work that I do in the systems biology field is an in silico analysis of the metabolism happening in living organisms. So that means I used to develop efficient algorithms and data structures to model biological systems. So it's a computer representation of the biology. Now, the uniqueness is that I do not look at only a single part of metabolism. I look at the entire system as one entity that allows me to develop hypotheses and design experiments at the genome level. At the heart of all the systems biology work is metabolic model. So in the next slide, I'll give you a brief primer of how metabolic models are developed. This figure shows the central dogma of biology, which transfers information from DNA to RNA to protein and then to a reaction through transcription, translation, and catalysis. So that means if you have an annotated genome and then you march through it, at the end you will get a list of reactions that are possible to happen in that organism. So once we have that list, the next step is to ensure that all of these reactions are elementally and charge balanced. Because if there are imbalances present in your reactions, it invalidates all the predictions coming out of your model. In the next step, we develop an artificial reaction called the biomass reaction that describes the accumulation of cellular macromolecules like amino acids, DNA, RNA, lipids, etc. to a hypothetical component called biomass. This reaction is representative of cellular growth. So in this network structure, it is inherent that there will be gaps because we have lack of knowledge of our biochemical systems. So in the next step, we go back to literature. We do sequence similarity search with other taxonomically related organisms to fill in those gaps so that our network model is well connected. Once we have all of this at our disposal, we arrange them in a mathematical form called stoichiometric matrix, where we have rows of metabolites and columns of reactions. And each of the numbers in this matrix represent the stoichiometric coefficient of a particular metabolite in a particular reaction. So with this mathematical structure at our hand, we make a very important assumption that is called mass balance at pseudo steady state. That means the reactions happening in our systems are usually happening much faster compared to other biological processes like growth or transcription. So if we assume that the change in concentration of a particular metabolite I is in pseudo steady state, then it should go to zero. That gives us a system of linear equations, and that can be defined by this higher order cone shape. So any point in that higher order cone shape is a feasible solution for our system. Now, since this is an indeterminate system, we use different sorts of optimization tools to find out what is the optimal location of the flux distribution in the model. And one popular such tool is flux balance analysis, or FBA. FBA tries to maximize the rate of the biomass equation that I showed you earlier, subject to these mass balance constraints, as well as thermodynamic bounds on the reaction rates and environmental constraints like nutrient uptake limitations, etc. Now, in addition to flux balance analysis, there are a host of other tools that people have been using for the last couple of decades. And we have been using those to model all different types of living organisms. And I'll start with one single cellular organism, a microbial pathogen called Staphylococcus aureus. Staphylococcus is a serious human pathogen that causes a number of diseases in the human body. And one important aspect of staphylococcal metabolism is that it's highly antibiotic resistant, and that's why conventional drugs keep failing against it. So there is a serious need for developing novel therapeutics. As part of that mission, I developed a new genomical metabolic model of Staphylococcus aureus in collaboration with Professor Thomas at University of Nebraska Medical Center. To do that, I assembled the initial reaction set from the 3 previously published models of Staphylococcus. And then I went back to literature, as I showed in my earlier slide, to figure out what are the missing functions and bring them from the literature to our model. I ensured that all of those reactions are balanced. And in the next step, I checked that the model is thermodynamically feasible. Then I used a tool called Rumen to reconcile the discrepancy between model predicted and experimentally observed gene essentiality information. And last, I used condition-specific and mutant-specific regulation information to further define the model. Now, in the next few slides, I'll briefly explain the last 3 steps of this process. First, fixing thermodynamic infeasibilities in the model. Now, this issue arises from the principle of mass balance. What the principle of mass balance steady state tells us is that whatever gets in the system must get out of the system, right? So that means if the flux of reaction A is 10, the flux of reaction C will also have to be 10, but it doesn't tell anything about the other 3 reactions, right? So a situation like this can arise, right? Where the other 3 reactions can go into an infinite loop, which is not thermodynamically feasible at all. Now, to break this kind of cycles, we can use different approaches. In this diagram of the central metabolic pathway in Staphylococcus, I have highlighted the reactions in different colors. That denotes whatever strategy we have used. So one of them is removing reactions. And there can be different cases. I'll show them one by one. The first one, if there are duplicates present in the reaction set. So the strategy is to just remove one of the duplicate reactions, and that breaks the cycle. Another example is there can be lumped reactions where, in addition to all the individual steps of a particular pathway, there is also one reaction that connects the initial and the final step. So even if those reactions are biologically sensible, in terms of modeling, they don't make any sense, and we can just remove the lumped reaction. Another type of errors that can happen are one single metabolic conversion from one metabolite to other, but using multiple different cofactors. Now, if we have cofactor specific information for a particular organism, we can use that and remove the other reaction from that. So that also breaks this cycle. Now, in addition to removing reactions, we can also change the direction of some reactions. And here is one example where this is a cycle involving methylglyoxal, lactose glutathione, and lactate. But if we look at the delta G information for this particular reaction, we see that it is highly thermodynamically favorable, so we can make it an irreversible reaction. And in this case, it will also break the cycle. So those are different strategies how we can make our model thermodynamically feasible. The other type of refinements that we did was reconciliation of gene essentiality prediction between the model and the experimentally observed essential genes. Now, Matt was a very talented undergrad who worked with me in this project, and he is now at MIT as a PhD student. What Matt did is compared the essential gene set between the model prediction and the experimentally observed. And then once we do that, we find two different kinds of errors. The first one is NGG, where model is predicting a particular gene to be essential, which is actually not essential. That means our model is missing some reactions or genes that should have been there. The other type of error is GNG errors, where model is incorrectly predicting essential gene to be non-essential. So that means model has some extra functions or extra genes or reactions that should not have been present there. Now, to reconcile these errors, Matt used a tool called ProMatch that tries to add functions or remove functions based on biochemical evidence in literature and also sequence similarity search. And then he reduced those numbers to some extent, but this is all we could do at that point, given the data at our disposal. Here is one example of how ProMatch works. So in this linear pathway that leads to nucleotide metabolism, this is an essential pathway for Staphylococcus. And the green highlighted reactions, glucose 6-phosphate isomerase and ribose phosphatidylimerase, those are GNG genes because they are essential, but our model is not predicting them to be essential because there is an alternate pathway, right? So we went back to the literature and found that this reaction may not be happening in this direction in Staphylococcus. So that gives us confidence to remove that direction of that reaction, and that makes these two green reactions essential. So that's one example of how ProMatch can reconcile the discrepancy between model prediction and experimental observation. Now, for these two sets of examples, we used information already published in literature and in different databases, but we also did our own experiment. Professor Thomas's lab at UNMC did measured excreted metabolite rates for 8 mutants in the central carbon and nitrogen metabolism. These mutants are highlighted in yellow in this pathway. They play important role in many important pathways in STAPH, including electron transport chain, energy metabolism, PCA cycle, etc. So we chose these metabolites and we measured the excretion rate of those different metabolites in chemically defined medium with glucose and without glucose. Now, we can simulate the model and then match the prediction from this experimental data. And to do that, I used condition-specific and mutant-specific regulations. For example, MEND is a mutant that simulates anaerobic growth profile, and it represses oxidative respiration. So in MEND mutant, we suppress the respiration pathways and tries to match the metabolite excretion rates observed from the experiment. For media-specific regulations, we know that when glucose is present in the growth medium, carbon catabolic depression is one of the major depression that happens. So we impose that depression in our models to match the profile of chemically different medium with glucose. Now, with this refined and curated metabolic model at our hand, going forward, we wanted to answer the question of how mutants of Staphylococcus adapts to different growth scenarios and still grows when they were not supposed to grow. And we went back to the same set of mutants and we viewed them in different conditions and collected data from different growth phases, did metabolomics and transcriptomics experiments, and obviously found differentially abundant metabolites and differentially expressed genes. Now, with these results, what I'll be doing is employing two different optimization algorithms. The first one tries to constrain the reaction rates with our experimentally observed gene expression data as well as thermodynamic information. The idea is, if a reaction is thermodynamically very favorable, it will require less enzymes to occur at the same rate compared to another reaction that is thermodynamically not that favorable. So doing that will give us a mutant flux profile that is consistent with both gene expression data as well as thermodynamic information. And we also have the metabolomics data at our hand, which we can use to validate the predictions from this algorithm, right? In the next step, I'll use this optimization framework that will try to find the minimum number of reactions that must have been regulated in the different mutants so that the observed mutant phenotype that we found in the first optimization solution matches. So this will give us important regulatory points in the metabolism that we can target for designing new therapeutics. Now, at this point, from single cellular organism, I'll switch to multicellular organism or eukaryotes, starting with a plant, rice. Rice is the most abundant food crop in the world, feeds more than half of world's population. What we know is that elevated temperatures at early developmental stage of rice adversely affect the seed size, nutrition content, and yield. So it is a question of high concern since the global warming effect and everything is increasing the average temperature of Earth. So we wanted to ask these questions. Is there, or if there is, what is the global regulatory mechanism that is happening when rice seed is subjected to elevated temperatures? And if any sort of omics experiment can help answer that question. Now, Professor Walia's lab did just that. They did a temporal transcriptomic analysis of rice seed at the early developmental stage. And from that, I did a differential gene expression analysis to find what are the genes that are most responsive to heat stress. From those set of genes, I did a co-expression analysis to find which genes are mostly co-expressed with the most number of genes. And we call them quote-unquote potential hub genes that are our candidates for global regulators. Then I developed and implemented a tool called MiReN, which I'll explain in the next slide, that finds the minimal regulatory relationship for a particular gene of interest. In this slide, I'm going to show how MiReN works. So MiReN tries to connect the change in gene expression level with the expression level of regulators for every gene in our organism. And the way it connects those two is with an interaction coefficient, which basically tells us whether this interaction is an activation or regression, depending on the positive and negative signs and the magnitude. Now, this set of constraints has to satisfy so that I can minimize the discrepancy of experimental gene expression data and the predicted gene expression from these equations. And subject to all this, I try to minimize the number of regulatory connections on a particular gene. So that means this algorithm will try to rank the regulatory influence on a particular gene of all the other genes in the network. And then we can take the, depending on what we want to do, top 1% or top 2% as the most potential or most influential global regulators. Now, using MiReN, we predicted a regulatory network for a lot of different genes. And in this slide, I'm showing two known regulatory networks where I'm trying to say, okay, MiReN could capture the already known regulatory influences currently. The first one on the top is slender rice 1, which is a growth hormone, and it is known to repress a lot of genes in the gibberellic acid response pathway, which is responsible for plant growth and stem structure. Now, MiReN also predicted that they're all SLR1 represses all of those genes. The second one, XL21, is an immunoresponse gene, and it upregulates a lot of SNARP1 and WRKY genes, which are responsible for pathogen resistance and immunoresponse. And we see that MiReN predicts that XL21 upregulates most of the genes. Now, these were already known regulatory networks, but we use MiReN to also make new predictions. For example, we picked the cell division cycle gene WD40, and it has a lot of different important roles in rice, especially in seed development and also in stress response. What MiReN predicted is that WD40 upregulates a lot of different genes, and many of them are important transcription factors in rice, which play very important roles in gene expression depending on different abiotic stresses as well as growth and development of the seed. So these results are very important because it gives us an idea of if this global regulator like WD40 is upregulated or downregulated, what happens downstream? Now, in this slide and the previous slide, I showed regulatory network results where I know the regulator. I want to know what are the potential genes downstream that are regulated. But MiReN can also work backwards. If we have a group of genes that are of our interest, we can answer this question. Okay, what are the most potential regulators for those genes of interest? That's what we did for example, the PDAC transcription factor family. In this diagram, the yellow dots in the center of the star diagrams are the members of the PDAC transcription factor family group. And all the other genes in light blue, they are the MiReN-predicted potential regulators for those particular genes. So from these three slides, what we could gather is that MiReN is flexible. It can work either top down, where I know a regulator and I want to find what are the genes that are regulated, or I know the genes of interest, I want to know what are the potential regulators. Also, in this study, we used MiReN with transcriptomic data, but MiReN can work with any level of omics data. It could be proteomics or metabolomics. So going forward, Leila is a senior in our department. What she's trying to do is incorporate this omics data on top of a rice seed metabolic model to find out what are the phenotypic changes happening in the reaction level. So to do that, she is using an already published rice leaf model and then customizing and curating it for rice seed, which we call the best seed model. And then she is employing the state control and stress data on top of that model to find the two context-specific models. And then studying what are the phenotypic behavior changes during its stress, and if we can develop a new hypothesis from the reactome study that will lead us to develop a stress-tolerant rice variety. Now, moving from rice, let's go to human, and the problem I'm focusing on is pancreatic ductal adenocarcinoma, or PDAC. Andrea is here. She's a very talented undergrad. She has been working with me on this project for the last couple of months. And the reason why we were interested in this project is that pancreatic ductal carcinoma, although being very rare, it has a very low survival rate. And the problem, the reason why is that they are very hard to diagnose, and most of the people are diagnosed at a very late stage. And also, PDAC is very resistant to different kinds of therapies. So what we thought is that we could do a reactome level study to see if we can find any significant metabolic behavioral change that will tell us to find new targets to develop or use different drugs. And to that end, we also had a lot of different omics data at our disposal, and we went to one of them, the Cancer Genome Atlas, or TCGA. We collected transcriptomic data on healthy and cancerous pancreas cells, and then we used the global human metabolic model, which is basically a consensus model of all different tissue types in humans, to customize a model for the human pancreas. We used a tool called Integrative Metabolic Analysis Tool, or IMAT, which basically tries to customize or tailor a model based on upregulation or downregulation of gene expression data, and also a given objective function. So we developed two models, one for the healthy pancreas, one for the PDAC cell, and compared those two models. And the comparison happened in terms of comparing the maximum and minimum possible flux space for each and every reaction in the healthy model and the cancerous model. And when we take that information to the pathway level, we can see which are the pathways that have most of the reactions either upregulated or most of the reactions downregulated. And this is the result, but we'll focus on some of them because some of them are already known to us. These are the ones that are unique to this study. Now, in the next slide, I'll explain them in more detail. So in this diagram, I'm showing my results in a summary, and I'll go through my results one by one. So most of the metabolic reprogramming that happens in PDAC cells are driven by an oncogene called KRAS, which is known to upregulate all of the small sugar uptake pathways, glycolytic pathways, pentose pathways, and hexosamine biosynthesis pathways. So this is a known information. What was unique in this study is that we see there is a lot of acetate present in the PDAC cell because of high flux through the glycolysis and pyruvate those pathways. Also, PDAC is surrounded by fibroblast cells that produce a lot of acetate, and the cancer cell can uptake those acetate. So that gives us a lot of acyl-CoA, and that drives the TCA cycle as well as the fatty acid biosynthesis pathway. So basically, supplying the major fuel source for cancer cell proliferation. The other important observation was that the cancer cell tries to minimize the oxidative stress as much as possible. So the glutathione reductase pathway is upregulated in PDAC, which tries to consume the hydrogen peroxide that is generated as quickly as possible. So obviously, if a cell is trying to grow rapidly, it will try to minimize oxidative stress. Another observation which was very interesting to us was while the mitochondrial fatty acid beta oxidation pathway is upregulated, the paroxysomal fatty acid beta oxidation pathway was not. The reason is that the mitochondrial pathway, in the mitochondrial pathway, most of the acyl-CoA molecules that are oxidized are very small chain, and they produce ATP. So obviously, a rapidly growing cell will favor a quick turnover of acyl-CoA molecules, and also use that ATP for driving the energy required for other processes, right? While in the paroxysomal pathway, it doesn't produce any ATP. It oxidizes very long chain acyl-CoA molecules, and it also generates hydrogen peroxide, which increases the oxidative stress. So it makes sense that the PDAC cell will most likely downregulate that pathway so that it doesn't carry the burden of all these long chain molecules, which doesn't produce any energy and also produce increased oxidative stress, right? Also, we see that the membrane phospholipids are degraded at a faster rate, which probably means that since this is a rapidly proliferating cell, it will try to recycle all those membrane phospholipids for making the new cells. So with all these observations, we thought that, okay, we know where are the most significant changes happening. Can we see if there are drugs associated with those genes or those pathways? And if we can recycle or repurpose some of the already used drugs in that purpose. So that's what we did. In this diagram, on the top level, I'm showing all of the different drugs that are associated with these genes that are upregulated in PDAC. And there are a lot of associations going on, and the drugs are colored depending on their approval status. But what we also did is check which of these genes are most growth coupled in PDAC or have the most effect on the growth rate. And the first one that came out was SLC2A1, which is associated with the transport of small sugar molecules, not only cross-membrane, but also between interorganelles inside the cell. And in our model simulations, this became actually an essential gene. That means it is the most growth rate limiting factor. So this could be one of the most potential targets. There are other three which I will not be explaining for the sake of time, but they also have important roles in cancer cell proliferation. Now, so these were my research on eukaryotic organisms. Now I'll move to multi-organism systems or microbial communities. They are everywhere. I don't have to explain this slide to all of you. They are playing important roles in natural processes and artificial processes, and we use them to our benefit in a lot of different purposes, including human health and disease and also artificial processes like biological wastewater treatment. Now, the way we define microbial community is slightly different. To be a community, it has to be at least two different species, and they have to talk to each other. Now, this talking is in terms of exchanging of biochemical fields like metabolites. And these interactions can be different types. There are positive interactions where one or both of the organism is benefiting from the interaction. There can be negative interactions where one or both of the organisms are suffering from the interaction. Now, what happens in most of the naturally operating microbial ecosystems is that there is never one single type of interaction present. There is always a complex combination of them, and they're very difficult to assess in vivo. So we can use models of different species and try to see what are the interactions and how that interaction dynamics change if we change the environment. And for simulating microbial communities, the tool that I have used the most is called d-OptCom. It was developed in Professor Costas Moranes's lab at Penn State, And what OptCom tries to do is connect the individual flux balance analysis problem for each of the microbial species with something called an interorganism flow constraint. This is basically a set of mathematical equations that describe the exchange of metabolites between different species. And subject to all this constraint, OptCom tries to maximize or minimize the community level of function, which can be either the growth of the entire community or bioengineering objective if this is an artificial community. The advantage of OptCom is that it can capture the trade-off between the species level fitness function in the flux balance analysis and the community level fitness criteria. Also, OptCom can accommodate any number of species because it just increases linearly, and it can accommodate any kinds of positive or negative interaction. Now, using OptCom and some other tools that I'll show later, I simulated two different microbial ecosystems, and I'll start by the rumen microbiome Virome ecosystem. Rumen is the first step in cattle digestion process. So whatever the cattle is eating goes to rumen first, and it harbors a diverse array of microbes, including bacteria, fungi, archaea, and viruses. Now, there are a lot of known information about the microbial interactions, but the role of the virome is not well defined. One hypothesis is that the viruses inject some of their metabolic functions into the microbial hosts, and that enhances or changes their metabolism to some extent. So our goal was to find out what role these viral auxiliary metabolic genes that are injected to the bacterial host is playing. So what we did, we chose three major functional genes in the rumen: bacteroids, firmicutes, and archaea. And then selected three representative species: Rumenococcus, Prevotella, and Methanobrevibacter, as the representative of those three major functional genes. And then we asked this question: what are the unknown interaction between these three members, and what role does the virome play in modulating those interactions? To find the unknown interaction, we utilized different tools, starting with this pair called GapFind and GapFill. What GapFind does is finding gaps in the metabolic network of an organism. Let's call it the host species. And GapFill tries to fill in that gap from borrowing function from another species or a database. And each of those reactions that we add, we call them GapFill solutions. So our step-by-step procedure was to check each of those GapFill solutions against different criteria. The first one was whether it's a transport mechanism or not. If it's not a transport mechanism, that means it's an internal reaction. And if it's an internal reaction, then we check whether it is present in any taxonomically closely related organism or not. If it's not, then it's highly unlikely that the reaction should be put in that host state. So we reject the solution. If it's present in other closely related organism, then we check if addition of the reaction would create any thermodynamic infeasibility, like I showed before. And then either accept or reject the solution. What is interesting is that when it's a transport mechanism and we see that the host species has a transporter annotated for it, we hypothesize that it might be a potential interaction between the donor species and the host species. So the idea is that complementation of metabolic functions give rise to interaction. So using this step-by-step procedure, we found some de novo interactions that are happening between the three species of our rumen microbial ecosystem. Now, in the white font, you see the already known metabolic transactions between the three species, but we found 20 new interactions of different metabolites, including small sugar molecules, vitamins, cofactors, and amino acids. Some of these findings are very expected. For example, Rumenococcus is a complex carbohydrate degrader. So it is expected that it will supply small sugar molecules to the other members. That's what you see. But some of them are very interesting as well. For example, Methanobrevibacter is supplying vitamins and cofactors, which was not known before. So once we had a good idea of what are the metabolite transports between these three species, we shifted our focus to the viruses. First, we found what are the viral species that are associated with the microbial host. I downloaded the proteome, did a sequencing research against NCBI non-redundant database, and found what are the potential reactions or genes that we can include as viral auxiliary metabolic genes into the bacterial process. Once we did that, we saw a wholesale change in the metabolism of the microbial species. On average, around 11% of the reactions have increased their flux space because now the models have more functionalities. So in this star diagram, the lightly shaded portion is the flux space after the viral auxiliary metabolic genes were added, and the dark portion is before. So in all of the models, you see that the lighter shaded portion is bigger than the darker shaded portion. So if we go to specific results, we see that in Rumenococcus, it utilizes sugars in a faster rate now, and also it has a high flux to pentaphosphate pathway. In Prevotella, we see high energy metabolism. In methanolravector, we see that it has now adapted to dextrose sugar more instead of pentose and hexose sugars. And also carbon flux has shifted from glycolysis to pentaphosphate. Now, how does that translate to the interactions between these species? We did our step-by-step procedure again and found what are the interactions. And we see that the transport of mannose and sucrose between Rumenococcus and methanolravector is not active anymore. So that means methanolravector is now less reliant on the sugars produced by Rumenococcus. We also see that although these molecules are produced at a higher rate by methanolravector and Prevotella, they are not consumed as much. That means these metabolites are in surplus in this moment, right? So that gives us these ideas that using the viral auxiliary metabolic genes results in higher utilization of the feed molecules and also more production of energy and other nutrients, less reliant metabolism between the microbial species. That means they are now more robust. Also, since there are surplus nutrients, this can be now absorbed by the cattle host, which improves the energy and nutrition of the cattle host. So this is where this project ends, and then I'll focus on another community work where we looked at the freshwater lake methanotrophic ecosystem. This was a combined work of me and two other very talented students, Tony and Shardar, this year, and they are now MD students in different places. So, like any freshwater lake in the world, Lake Washington harbors methanotrophs and methanogens, and they are community dynamics chains depending on the methane oxygen counter gradient in the anoxic sublayer. Now, as a first step in modeling this four-member ecosystem, we simulated a community of two methanotrophs, Methylobacter and Methylomonas. They are in a competitive relationship for the carbon source methane, and they also compete for oxygen and nitrogen. But in addition to that, we see Methylomonas sometimes produce formaldehyde, which is toxic to Methylomonas. And then Methylobacter has the ability to take up that formaldehyde and metabolize it. So this establishes a kind of synergistic relationship between these two organisms. Now, since there is an oxygen methane counter gradient present in the lake water, we simulated this community in the gradient of oxygen and methane. And obviously, we see that when it's a nutrient-rich environment, the total community biomass is high. But what is interesting is that when it's a nutrient-scarce scenario, we see Methylobacter is not dominant community member. But when it's a nutrient-rich scenario, Methylomonas becomes the dominant community member. So that gives us some idea about the metabolic efficiency or the feed utilization efficiency between two organisms. Now, if we look closer and look at two different conditions, one is nutrient-rich versus another oxygen-deprived condition, we see that in a nutrient-rich condition, Methylomonas is taking up most of the methane, but the thickness of the lines represent the magnitude of reaction flux. So Methylomonas is taking up most of the nutrients, and obviously it is growing at a higher rate, as I showed in the previous slide. And interestingly, it excretes many of the, a lot of the carbon that it has taken up as methane. It is excreting a lot of that in the environment as carbon dioxide. So it's not accumulating all of that into its biomass. But what happens in the oxygen limited condition is that there is a stricter competition between these two members now. They are both growing at a similar rate. And also, most interestingly, Methylomonas is not excreting the carbon dioxide anymore. Rather, whatever carbon dioxide is produced in the PCA cycle it is recycling that carbon dioxide back by reversing the direction of the pyruvate carboxylase reaction here. Now, there is no solid evidence for this, but in many other organisms, which are methanotrophs and methanogens, we see that pyruvate decarboxylase is a highly oxygen-sensitive enzyme. So that might be a possible reason why this reversal happens. But this also gives us an interesting aspect of how the community dynamics is driven by the methane-oxygen counter gradient. So with all these projects, what I try to show you is that if we have good mathematical models, this will allow us to a very decent analysis of metabolism of any living system, be it single organism system or multi-organism systems. And once curated and refined, these models allow us to do a lot of different things. We can find novel therapeutics, we can infer regulatory information, we can decipher if there are complex interactions between species in a microbial community. And together all of these, it allows us to discover and redesign the metabolism of microbes, plants, human, and microbial ecosystems. And that is the goal of my future research. I will develop and use computational tools to understand and then engineer these living systems for the benefit of humankind. There can be different directions towards natural and artificial microbial communities to understand their dynamics better, so that I can either engineer them for increasing the efficiency of commercial process or addressing human health and disease issues. Also, I look at extremozymes in nature. Some of the commercially important enzymes are found in extreme environments on Earth. For example, Arctic permafrost, even nuclear bioreactors. So I can take those enzymes in the lab, understand their metabolism, see if there are enzyme promiscuity present, and if I can use them for industrial biotech performance. On the systems biomedicine field, I want to look at multifactorial diseases like obesity, cancer, and diabetes, and to formulate a mathematical model of them, understand the disease dynamics and the multifactorial nature that will lead to developing novel therapeutics. Now, with all these ideas in my head, I'm going to join Professor Coppin at University of Virginia as a postdoctoral research associate very soon. And I'll look at network modeling of biological systems to predict drug targets and also understand the disease mechanisms. All these works that I have presented today was done in the Systems and Synthetic Biology lab led by Professor Saha. You already know that I have joined in 2016. I was the only student at that point. Now this family has grew so large with even grandkids, thanks to Wendy Brown, who is another graduate student. Now, this is an awesome group of people, and I feel myself very lucky to have them as my colleagues. Professor Saha has led all of these projects, and he has provided me guidance. He has allowed me to work on my ideas when he thought that they would work, and he has helped me in every aspect of the academic and personal life. So I'm greatly thankful to him. Also, I had some fantastic collaborators during my PhD, Professor Thomas at UNMC, Professor Walia, agronomy and horticulture, and Professor Fernando in animal science. And all of my projects benefited from their constant input and suggestions. In addition, I'm thankful to the lab, especially Dr. Cheryl Emerton, Dr. William Schroeder, who just became a doctor last week, and Adil Al-Silvi, as well as all of the undergrads that have worked with me. My fundings came from Nebraska System Science Initiative, Professor Saha's Faculty Startup Grant, and Nebraska Collaboration Initiative. With that, I'd like to conclude my talk, and thank you for listening. I'd be happy to answer any questions you might have.